So before we begin our overview of protein synthesis, let's first explain what all these things represent. These are all the basic base pairings. T goes with A goes with U, and G goes with C, and vice versa. DNA is in a double helix shape, and this represents RNA polymerase, which will be unwinding the DNA, which will eventually end at the termination region, which is going to be represented by this. So beginning in the nucleus of the cell, we start with transcription. Transcription begins with initiation, which continues along with elongation and ends with termination. In initiation, the RNA polymerase attaches to a promoter region on the DNA and begins to unzip the DNA into two strands. A promoter region for the mRNA transcription often contains the sequence TATA, called the TATA box. Elongation occurs as the RNA polymerase unzips the DNA and assembles RNA nucleotides using one strand of the DNA as a template. As in DNA replication, elongation of the RNA molecule occurs in the 5-3 direction. In contrast to DNA replication, new nucleotides are RNA nucleotides rather than DNA nucleotides, and only one DNA strand is transcribed. Termination occurs when the RNA polymerase reaches a special sequence of nucleotides that serve as a termination point. In eukaryotes, after transcription, but before an mRNA molecule leaves the nucleus, it undergoes mRNA processing, where the following alterations take place. A 5' is added to the 5' end of an mRNA. The 5' is a guanine nucleotide with two additional phosphate groups, forming GTP. Capping provides stability to the mRNA and a point of attachment for the small subunit of the ribosome. Next, a poly A tail is attached to the 3' end of the mRNA. The tail consists of about 200 adenine nucleotides. It provides stability to the mRNA and also appears to control the movement of the mRNA across the nuclear envelope. Next, RNA splicing removes nucleotide segments from mRNA. A transcribed DNA segment contains two kinds of sequences: exons, which are sequences that express a code for a polypeptide, and introns, intervening sequences that are non-coding. The original unprocessed mRNA transcript contains both the coding and non-coding sequences. Before the mRNA moves to the cytoplasm, small nuclear ribonucleoproteins, or snRNPs, delete the introns and splice the exons. Lastly, alternative splicing allows different mRNAs to be generated from the same RNA transcript. By selectively removing different parts of the RNA transcript, different mRNAs can be produced, each coding for a different protein product. Now we're at the final stage of protein synthesis, translation. Translation is a process by which the mRNA sequence is converted into an amino acid sequence. It occurs in the ribosome. Amino acids present in the cytoplasm are carried by the tRNA molecules to the codons of the mRNA strand at the ribosome according to the base pairing rules A with U and C with G. Some tRNA molecules can bind to two or more different codons. For example, codons UCU, UCC, UCA, and UCG all code for a single amino acid, serine.